What if we collect all the coronavirus from this world and store it in a place? How much space it would acquire? A room? A box? Not at all. It just fits in a teaspoon. This was estimated by Australian mathematician Matt Parker. It's surprising how this teaspoonful of virus took millions of lives. In this video, let us understand how vaccine works on this deadly virus. Before talking about the vaccine, let us understand what's the need of this vaccine. Why we are in need of taking this. Let's talk from the basics. When coronavirus enters our body, it makes use of our own cells to replicate itself and makes millions of copies inside our body. Our body is made of trillions of cells. When this coronavirus enters our body, it cannot directly enter any cell because cells are covered by cell membranes. This cell membrane borders our whole cell. For this instance, understand a cell as a room and cell membrane is its wall. As you cannot enter a room through a wall, you must find doors. In the same way, coronavirus can't cross this wall and it must find doors. In our cell membrane, there are many receptors for different functions. Coronavirus makes use of one of these receptors and gets into the human cell. This virus have spike proteins on their wall that attaches to these receptors and makes its way into our cell. So our scientists worked through all these and thought, what if we attach antibodies to these viral spikes? Then virus cannot bind to our receptors and can't enter our cells. These antibodies must be developed by our immune system. Thus, our main aim now is to produce these antibodies at right time in right amount so that virus cannot enter our cell. To understand further, we need to look at the structure of this coronavirus. This virus is surrounded by spike proteins on their wall. Inside the wall is its genetic material. Basically, genetic material guides how to proceed in each step. So scientists took extensive studies in reading COVID genetic material. As soon as they completed this process, they made it public. All the biotech companies stopped their independent projects and started studying genomic structure of this virus. So basically, all the companies took the data from the genome of COVID and converted them into mRNA. mRNA meaning messenger RNA. Let me break this down to you. Take us as an example. We have DNA in each cell of our body. This DNA is converted into RNA that in turn converts into proteins. The conversion process are more complex and let us cover that in another video. These proteins are responsible for all our actions and physical structures. The same DNA test is done when the baby matches the characteristics of your neighbor. Coming back to corona, these biotech companies took the mRNA into equipped laboratories and scientists put specific mRNA into nanoparticles. When these nanoparticles are introduced into our body, they act as transporters. They transport desired mRNA into our cell. Now it's up to our cells to make proteins from these mRNA. Ribosomes in our cell take these mRNA inside, read them and make them a particular protein by adding amino acids. Generally, whenever a cell makes a protein, it shows them on their outer molecules. In the same way, these newly synthesized proteins are shown by the cells. And like this, our immune system comes to know about this newly synthesized protein. The molecule that shows this protein is called MHC class molecules. As our cell makes many copies of this protein, at least one must go and attach to MHC class molecules and others are secreted out of the cell. There are several types of immune cells in our body and out of these, cytotoxic T cells come and attach on MHC where these new proteins are present. And other proteins that are released out come in contact with B cells and macrophages. The macrophages eat these proteins and show some of its pieces upon them and makes the contact of these pieces with helper T cells. The helper T cells reads these pieces and release cytokines. These cytokines are some sort of orders that tells cytotoxic T cells to produce granzymes and memory T cells. Granzymes are useful in killing infected cells. The other cytokines guides B cells to produce plasma cells and memory B cells. Plasma cells release antibodies that binds with the spike proteins and inactivate this virus. When actual coronavirus attacks our body, granzymes kill infected cells. Memory B and T cells remembers the old attack that is actually caused by mRNA vaccine, and this time these cells activate plasma cells to release antibodies. These antibodies bind with spike proteins of corona and inhibit them from binding to actual cells. This type of mechanism or mRNA vaccination is incorporated by Moderna and Pfizer companies that are actually running successful vaccination all over the world. But the vaccine from our Indian company, that is Bharat Biotech, is different. Bharat Biotech have inactivated the coronavirus itself and injected them upon us. And thus our immune system works its way through this and memory cells remembers the same. When real infection occurs, they fight on right time and kill corona before it enters our cell. And that's how mRNA vaccines and vaccine developed by Indian scientists work. And that finishes this video. If you did really learn something, then share this video with your friends and family. And of course, your subscription inspires me to make more videos like this. So don't forget to subscribe. Thank you.